Assalamu alaikum, dear students. Today we will continue with the process of photosynthesis, and we have to study about the mechanism of photosynthesis. You know that the mechanism of photosynthesis can be understood under two important phases. One phase is the light reaction and another phase is the dark reaction. So first of all, we will provide an overview of the two phases of the photosynthesis, that is the light reaction and the dark reaction phases. So when we talk of the light reaction is also called as light dependent phase of photosynthesis, or it can also be called as uh, photochemical phase of photosynthesis. This is because it is dependent completely upon the presence of light, and it occurs in the grana. If light is present, the light reaction can occur. If the light is not available, then the light reaction cannot occur. And what happens actually in the light reaction is that the photosynthetic pigments trap the solar radiation and convert it into the assimilatory power, that is ATP plus NADPH plus H positive. And this assimilatory power thus is generated in the thylakoid membranes. This assimilatory power is then utilized in the second phase of the photosynthesis, and that you call as dark reaction. And in that dark reaction, this carbon dioxide gas is fixed into the photosynthetic reactions to produce the sugar, and for that purpose this assimilatory power is a very essential requirement. So this was an overview of the two phases of photosynthesis, where you can see that the two phases are connected through the expenditure of what I mean to say assimilatory power in the second phase that was produced in the first phase. So now let us take a look upon the mechanism of light reaction. Light reaction you already call as photochemical phase. Photochemical phase of the photosynthesis, or you can call it as light dependent phase. Light dependent phase of the photosynthesis. This occurs inside the grana, or we can call as it occurs in the thylakoid membranes. Thylakoid membranes. So presence of light is essential. Light must be present. And when light is present, the photosynthetic pigment is present in the grana or in the thylakoid membranes can trap the solar energy, and that solar energy will be then eventually converted into the assimilatory power. So let us consider here the light, which is incident. Light is incident upon the photosynthetic pigments, and in these pigments the light is converted into ATP plus NADPH plus H positive. NADPH plus H positive. ATP is adenosine triphosphate, whereas this NADPH plus H positive is a reduced coenzyme, namely nicotinamide adenine dinucleotide phosphate. And separately this will be called as reducing power. Reducing power, and these two will be together called as assimilatory power. Assimilatory power. So assimilatory power will be generated in the light reaction, and then it will be utilized in the dark reaction phase of the photosynthesis. So let us see what actually is the mechanism associated with occurrence of this light reaction in the grana or thylakoid membranes. The mechanism of light reaction can be understood under two important headings, and these headings are the two pigment systems and the photophosphorylation. About the two pigment systems, I have already told you that there are two pigment systems, pigment system first and second, and each has its own reaction center and each has its own role to play in the process of photosynthesis. So this light reaction, its mechanism can be understood under two headings. One heading you call as two pigment systems. Two pigment systems. Number one, and number second you call as photophosphorylation. Photophosphorylation. So pigment systems, PS first with the reaction center P700, and then PS second with the reaction center P680. You have to recall that there occurs a manganese protein also associated with pigment system second that makes the oxygen evolving complex in the pigment system second. And the job of that oxygen evolving complex is to cause photolysis of water and to liberate the oxygen, the protons and the electrons. So this is about the pigment systems, and they will participate in the process of this light reaction. Now, photophosphorylation is another heading in the light reaction step. Photophosphorylation. Phosphorylation. Basically, phosphorylation is combination of adenosine diphosphate with inorganic phosphate to form adenosine triphosphate. And if this phosphorylation occurs in the chloroplasts in the presence of light, then we call it as photophosphorylation. So, photophosphorylation mechanism, it was explained for the first time to us by Arnon et al. In 1954. And this photophosphorylation, as you know, it is the combination of adenosine diphosphate with inorganic phosphate to generate ATP. And if this ATP formation from ADP and IP is driven by the light in the chloroplast, you will call it as photophosphorylation. Okay, photophosphorylation is of two types. In the thylakoid membranes, it has two types, or rather two mechanisms. One type you call as non-cyclic photophosphorylation. Non-cyclic photosynthesis. It is one of the types of phosphorylation which is also referred to as Z-scheme. And the second type of it is the cyclic photophosphorylation. Cyclic phosphorylation. So these are two types of phosphorylation, cyclic and non-cyclic. Non-cyclic photophosphorylation is very, very important. And it is actually much more efficient type of photophosphorylation than the cyclic one. So first of all, we will study about the non-cyclic photophosphorylation. But before going to the details of non-cyclic photophosphorylation, we have to first of all understand the primary reaction of photosynthesis or primary photochemical event. That is very important. Once we understand the primary reaction of photosynthesis, we can be in a position to understand the whole mechanism underlying this Z scheme or non-cyclic photophosphorylation. So before going to the details of this Z scheme, let us understand the primary reaction of photosynthesis. Primary reaction of photosynthesis. Primary reaction of photosynthesis. Or you can also call it as primary photochemical event. Photochemical event. For that purpose, we will consider the pigment system second. And you know that pigment system second has a reaction center P680. Then there are core molecules. Then antenna molecules are there. And a protein called as metallic protein or manganese protein is here, which is responsible for photolysis of water. So this is the pigment system second. And when light is incident upon this pigment system second, I already explained this thing that the light photons will be absorbed first by antenna molecules, then they will hand over the energy to the core molecules by means of a phenomenon called Forster transfer or resonance transfer. Then the core molecules will ultimately pass the energy to the reaction center, and reaction center, getting a sufficient quantum of the light energy, is in a position to emit the electrons. So we have to this time deal with this reaction center P680. And I have already told you that this reaction center P680 is a special type of chlorophyll A molecule, and this chlorophyll A molecule is already present in its ground state. And in the ground state, suppose it has two electrons. One electron is this and it is another electron. And both the electrons lie in its lower energy orbital. Lower energy orbital. Whereas this is the higher energy orbital. This is empty, because both the electrons are present in its lower energy orbital. So this is in the ground state. The chlorophyll molecule or its constituent atoms, we can say they are in the ground state. So in the ground state, this chlorophyll or P680 pigment molecule, it is a poor reductant and a poor oxidant. So I will write the properties of this. It is poor reductant or reducing agent, reductant, and it is a poor oxidant, poor oxidizing agent. Now we expect that this P680, it has to emit the electrons. So when it will emit the electrons, that means it has to act as a reducing substance or reductant, because it will reduce the primary acceptor. That substance which will first accept the electrons from this P680, that will get reduced, and this will act as reductant. But in the ground state, In the ground state, this P680 is a poor reductant. So we here say that it is a poor reductant. Why poor reductant? Because both the electrons are present in its lower energy orbital, and if we want to eliminate an electron from it or eject an electron from it, it is not easy. It will be rather difficult. Okay, now this difficulty is solved by the light. This is where the light plays its role. When this poor reductant P680 becomes excited by gaining a sufficient quantum of light energy, one of the electrons of it become excited to the higher energy orbital. So this is now the excited P680. Now its properties are changed. Now it will act as a strong reductant. Strong reductant. This is because if you want to eject an electron from this pigment molecule, you can easily eject it because it is already elevated to what? Higher energy orbital. And to give the electron from such an orbital is very easy for the pigment molecule. So it has now become strong reductant, and it is also a strong oxidant. If you want an electron be accepted by this, it can now accept that electron to its lower energy orbital. So now it has become strong reductant as well as strong oxidant. Now when it has become a strong reductant, it can now easily give the electron to the primary acceptor. If that primary acceptor is here available, it will give the electron which is in its higher energy orbital. It will be accepted readily by the primary acceptor. And we will at that time say that the primary acceptor has been reduced by this, and this has been oxidized. The pigment system, the reaction center P680 has been oxidized. But when P680 is oxidized, it will strongly attract an electron from manganese, which is present in its protein called as metallic or manganese protein. So it will attract that electron from what? Manganese. Manganese will provide the electron. And when manganese provides the electron, this manganese strongly attracts an electron from water. Here is water, a raw material in the process of photosynthesis. And water gets photolysed. And when water gets photolysed, it will furnish three things. One thing is oxygen that is liberated into the atmosphere. Another thing is the electrons, and still another thing is the protons. So this is suppose P680, and this is the manganese protein, and this is water. And you know that this is PS second. And in this PS second, what will happen? This water will split in the presence of light, because light is already there. There is chlorophyll and other pigments present, and there is presence of manganese divalent positive ions. There are some other, for example, calcium divalent positive, Cl negative ions. These are various catalytic ions which are present there. And this water will be now photolysed. It will be split into half of O2 plus 2 electrons plus 2 protons. And this reaction you call as photolysis of water. Photolysis of water. Okay, the photolysis of water I will show you in some detail here. Suppose in water we have two molecules of it which are being here utilized. Twice H2O gives us twice OH negative plus twice H positive. So here two hydroxyl ions are generated and two protons are generated from two molecules of water in the presence of light, then chlorophyll, and then ions, catalytic ions like manganese, Cl negative, calcium divalent positive, etc. Now these hydroxyl ions, they recombine and they will again produce water. Suppose they will generate H2O plus half of O2, half of O2 plus 2 electrons, 2 electrons. Okay, these are 2 electrons that are liberated here. This water again splits into OH negative plus H positive ions. Now take this OH negative, OH negative. This will give rise to hydrogen, H plus an electron plus half of O2. Now we have this hydrogen. Hydrogen will be split as H positive plus electron. H positive plus electron. Now let us see. Twice OH negative cut, H2O, H2O cut, OH negative cut, HH cut. Now we have net equation of this photolysis of water. That is twice H2O. On this side, we will here get 2 protons, 1 proton means 3 protons, and this proton, that is 4 protons, 4H positive. Then you have here 2 electrons, again 2 electrons, that is you have 2 electrons, sorry 4 electrons, 4 electrons. And in addition to this you have here half of O2 and this half of O2, that is the O2. So this is the photolysis of water. So this is about photolysis of water and about the primary photosynthetic reaction or primary photochemical event. I hope you have understood this much, and we will inshallah continue with the process of mechanism of photosynthesis. See you. Fi amanillah.